Welcome to MOOC Interactomics course. Today we are going to understand about another popular technique which is based on BioLayer Interferometry. It is highly useful for studying biomolecular interactions. BLI is based on simple dip and read system which is useful for measuring interaction between proteins, peptides, nucleic acids, small molecules and lipids. It is an optical technique that analyzes the interference pattern of white light reflected from two surfaces, a layer of immobilized protein on the biosensor tip and an internal reference layer. Any change in the number of molecules bound to the biosensor tip causes a shift in the interference pattern that can be measured in real time. Interactions are measured in a label-free environment with the ability to monitor binding specificity, kinetics, for example, rate of association and dissociation, and concentration analysis with precision and accuracy. Today we have Dr. Kaur with us from Paul Life Sciences, who will elaborate a little on the basics of BLI technology and its application in protein research.
Hello everyone, today I am going to give an introduction to BioLayer Interferometry and its application in protein research field. During the past 10 years, PhotoBio has developed a series of systems relying on the BioLayer Interferometry, which is a label-free technology and allows you to perform real-time analysis of biomolecule interactions and also to perform quantitation of the biomolecules in micro-volume sample sizes. Now let's have an understanding of the BioLayer Interferometry or BLI technology. Now let's have to understand how it works in the real life. The core part of the BioLayer Interferometry or BLI technology lies with the biosensor. If we take a close look at the biosensor, it actually made by optical fiber in the end. So at the very end of the biosensor, it is a two-dimensional biocompatible matrix. This matrix allows you to immobilize the molecules on this surface to allow further analysis of biomolecule interactions. The benefit of use this patented biocompatible matrix is that it's very uniform. It minimizes the molecule binding to the membrane, and also it is non-denaturing. So what happens when we are doing experiments with BioLayer interferometry? If we take a close look at the biosensor, we can actually observe two reflection layers. The internal reflection layer is engineered into the biosensor during the manufacturing process. And the second layer is an interface between the tip of the biosensor and the liquid. When the biosensor is put into the BLI instruments, the instruments will generate visible lights. These lights will go through the biosensor, and as we know, some of the lights will be reflected from the two layers of reflection surfaces. Since the white light has lights at different wavelengths, so the two reflected wave beams will interact with each other. Some lights at certain wavelengths will show a constructive interference. Some will show a partially constructive interference, and some will see negative interference or destructive interference. No matter what, the system will monitor the reflected two beams and plotted the composite or composed two wavelengths at this XY plot. Then by plotting the wavelengths or wave pattern of these different waves on this XY plot, we can get an original wave pattern. When an interaction happens, the molecule will bind to the end of the biosensor. And as a result, we'll see the secondary or second reflection surface actually moves down. And this moving down increased the distance between the two reflection surfaces. And this increase will change the interference between the two reflected waves. As a result, what we'll see is, originally we have wave pattern as shown here, but after the interaction happens, because the interference changes, so we'll see a different wave pattern. It's kind of shift from the original wave pattern. The distance of the shift is proportional to the size of molecules on the membrane, the amount of molecules bound in the membrane, and also the density of the molecules bound on the membrane. So the system will monitor this wave pattern shift in real time, and from there we can get understanding about the details of the biomolecule interactions. Now let's look at the example. If we are looking at the antibody-antigen interaction, as we know, the antibody will bind to antigen and form a complex. At the same time, the complex will go through a dissociation process and generate the antibody and antigen. As a result, if we have a biosensor which already have immobilized antibody on the very end or on the biomatrix, and if we put this biosensor into the solution contains antigen, the antigen will bind to the biosensor and result a shift of the wave pattern, which in turn generate the association curve between the antibody and antigen. Afterwards, if we move the biosensor to a buffer solution, the antigen will be dissociated from the antibody. Then from here, we can get a dissociation curve of the antibody-antigen interaction. So this is how the system works in the real time. Because the BLI technology is developed based on this biosensor, which can work with different sample types, it allowed us to analyze the interactions between biomolecules from molecules down to 150 Dalton to antibody, to recombinant protein, to virus, and to bacteria. However, this system may not work directly with the intact mammalian cells because of the size of the cells has exceeded the limit of the detection. And as another application, the BLI technology can work with different research purpose. We can use the technology to perform quantitation of the biomolecules. This can be achieved by immobilize a specific antibody or molecule onto the biosensor and use it to analyze the concentration of other molecules. It can also be used to analyze the kinetics of the biomolecule interactions for us to get a K-on, K-off, Kd. And in the pharmaceutical industry, it can be also used to perform functional testing, the epitope binding analysis, and also perform isotyping of the antibodies. In the next couple of slides, I'll give you some example about the application of the BLI technology in different fields. Firstly, let's look at the application of the BLI technology in kinetic application. When we're doing a kinetic experiment, we firstly need to coat the biosensor with a ligand. In this experiment, we are using the biosensors with streptavidin immobilized on it. So the ligand has already been biotinylated, and we'll first put the biosensor into the buffer to remove the protective agent from the biosensor. After that, the biosensor is moved into the wells that contains the ligand. The biotinylated ligand will bind to the streptavidin and generate a loading curve, as we can see here. Afterwards, the biosensor is then moved back to the buffer. Then we can get the baseline before the real interaction happens. As a fourth step of the interaction, the biosensor is moved into the wells which contains the analyte of interest. The analyte of interest will bind to the ligand, which has already been immobilized on the biosensor. And then from here, we can get the association curve between the analyte and the ligand. And as a last step of this analysis, the biosensor is moved back to the wells which contains a buffer. Then in the buffer wells, the analyte will dissociate from the ligand, and we can generate the dissociation curve. One thing we need to pay attention here is all the interactions, the curves, are generated in a real time. We can get many details from this analysis. Depends on the system you are using, we are allowed to analyze either 8 samples or 16 samples or even 96 samples at the same time, which dramatically speed up the analysis process. Now let's look at one of the publications which use the BioLayer Interferometry to analyze the binding between the two proteins. In this particular research, we are studying the binding of the MHC class 1 with another protein called CPXY203. So in this particular study, the researchers performed a very stringent analysis between the two molecule interactions. So they firstly used the SPR technology, which is another technology used to analyze biomolecule interactions. Then they get the KD value. And in another experiment, they then performed the analysis using the BLI technology. And the data shown here supports that the kinetic constants achieved on the BLI technology is the same as the one achieved with SPR technology. So the main purpose of this study is to show us that it is very important for us to validate our results in different testing platforms. So this will help us to be sure that results we get is not false positive. I believe this theory or this kind of thinking will be very useful when you are designing your own experiments. As another example of the application of the BioLayer Interferometry, let's look at quantitative analysis of BLI. When we are doing a quantitative analysis, the first step is to generate a standard curve. So during the setup, the biosensor is firstly deep into the wells, which contains the standards. The standard will bind to the biosensor, and by calculating the initial binding rate of the molecules to the biosensor, we can get a standard curve with a binding rate as a y-axis and the concentration of the molecules as the x-axis. After we get the standard curve, we can then regenerate the biosensors by putting the biosensors into the regeneration buffer followed by the normal buffer to neutralize the biosensor. Afterwards, the biosensors are dipped into the wells which contains the samples at unknown concentration. From the binding curve, we can also get the initial binding rate of these molecules at unknown concentration. And by plotting this initial binding rate onto the standard curve, we can get to know the concentration of these unknown samples. One advantage of using BI technology to analyze the concentration of the molecules is that it is able to analyze multiple samples at the same time. And also, it only takes you about 15 to 30 minutes to finish analyzing 96 samples. Depends on the assay you are using, you can actually regenerate the biosensors, which means that you only need 16 or 8 biosensors to analyze the whole 96 well plate or 384 well plates. So this dramatically facilitates the application of the BI technology in the pharmaceutical industry as well as in the academic research field. Here is one of the examples which shows that how we can use BI technology to detect the protein in a crude cell lysate. In this particular experiment, we actually analyze the samples with a system called BLITZ. The BLITZ is a single channel system developed using BI technology. So in the experiments, we are expressing 2 molecules or 2 proteins in E. coli. The research has shown that these 2 proteins will bind to each other and they can form a tight complex in E. coli. Between the 2 proteins, one of them has been His tagged. So we can actually use anti-His antibody to detect the particular fragment. We express these 2 proteins simultaneously in E. coli, and we run the cell lysate on a SDS page gel. From this gel, we can see that we actually cannot tell whether the 2 proteins are expressed in the E. coli or not, because there are very high background on the SDS page gel. So how can we do that? The researchers in this particular study used BLITZ platform to analyze the concentration, the expression of the molecules in the E. coli. What they have done is, from the cultured E. coli samples, they just take out 4 microliter of the cultured bacteria and then dip these 4 microliter samples onto the BLITZ system. Then we also use an anti-His biosensor attached to the BLITZ system, and the anti-His biosensor is then dipped into the sample, which is only 4 microliter. The His tag proteins, if they are present in the samples, they will bind to the His tag biosensor and generate a binding curve. So by looking at the generation of the binding curve, we can actually tell whether the protein of interest is expressed in the E. coli or not. So we can see here, by using only 4 microliter samples without any sample purification, we can easily perform the analysis to tell the presence of our protein of interest in the E. coli, which saves really a lot of time, from 30 seconds compared with 1 day of the Western blot. Now let's look at another application of the BI technology in the vaccine titer determination. As I mentioned earlier, because of the unique feature of the BI technology, we can actually use the technology to bind to the virus directly without further sample purification or process. Traditionally, in the vaccine industry, we are relying on SRID method to perform the quantitation of the vaccine virus titer. However, this method is very time consuming. It takes up to 3 days to analyze the virus titer. And also, it has very limited sample throughput, where it is only able to analyze maybe 15 or 16 samples at the same time. Thirdly, the sensitivity and accuracy of the SRID method is limited, and the interpretation of the final results is actually subject to personal interpretation. So this method may not be a very perfect method for you to determine the virus titer in a sample. People have also been using ELISA method to analyze the vaccine titer. However, similar to SRID, ELISA takes a very long time to finish the whole experiment process, and it has very low precision and to determine the titer of the virus, and has very limited dynamic range. To overcome all these shortcomings, the researchers are using the biotechnology to determine the virus titer in the real experiments. And as we can see here, from the sample preparation to the time we get the final results, it only takes about 3 hours for you to finish the whole process. I would like to show you how to use the biotechnology to select the right construct for protein crystallization studies. In these experiments, so the researchers would like to crystallize some constructs at different truncations. What they have done is they have attached the his tag together with the protein as a n-his tag protein. So the experiment is carried out based on 2 assumptions. Firstly, if the protein is folded correctly, the tag will be exposed on the surface, which allows it to bind to the biosensor. On the other hand, if the protein is not folded correctly, the tag will be buried inside the protein complex, and as a result, the misfolded protein are not able to bind to the biosensor with anti-his antibody coated on it. Also, the misfolded protein tends to form aggregates. So in this particular research, our researchers firstly perform a SEC size exclusion chromatography on a crude cell lysate. From there, they can get a pattern distribution of the different constructs. And also, by using SEC analysis, they can identify which portion is a soluble portion and which portion is the aggregated portion. As a second step of the study, the researchers then express the different constructs in the E. coli and use the BLI technology to analyze the binding of the expressed protein to the biosensors with anti-his antibody immobilized on it. So the researchers measured the initial binding rate of these constructs to the biosensor. And by calculating the initial binding rate, they can plot this data on this XY plot, as you can see on the upper right corner of the slide. It's very interesting to notice that the constructs with the highest initial binding rate actually corresponds to the constructs with the highest soluble portion. So these 2 constructs is selected for further crystallization study, and the researchers then get the crystals of these 2 constructs. The main learning point from this study is that by performing a 1-minute assay using BI technology on the Octet platform allows you to correctly prioritize the protein constructs which is suitable for downstream crystallization, which helps you to simplify your research process and speed up the process for you to get the crystals for further structural analysis. Because there are so wide applications of the biotechnology, we have developed a wide range of the biosensors for you to analyze antibody concentration or interaction, or you can use the biosensors to analyze the his tag proteins or GST tag proteins. Or if your protein is not tagged or they are not antibodies, you can also use other biosensors such as AR2G biosensors, which has amine group immobilized on the biosensor, or the streptoavidin biosensors, which includes the ACA biosensor, ACCA biosensor, and ACAX biosensor for you to analyze the streptoavidin or biotininated molecule interaction. So as a summary of my lecture, we kind of going through the basic mechanisms of the BI technology, and we also included some of the examples of using BI technology in different research fields, which including protein-protein interaction, protein quantitation, or even protein crystallization. The benefit of using the BI technology or even the Octet platform in your research field is that it allows you to perform your analysis in a short time. The platform is very straightforward and is very easy to use. Lastly, if you're working with the small molecule samples, this system allows you to analyze samples in DMSO up to 5% of the concentration. So here, I will wish that after going through this lecture, you have get a better understanding of the BLI technology, and also to understand how this technology will help you in your protein research. Thank you.
So today we have learned about the basics of BioLayer Interferometry technology and how it is used in different applications in research. In our next lecture, we will demonstrate the BLI technology to perform a case study of protein-protein interaction study. Thank you.